Welcome to the light-independent reactions of photosynthesis. Here's the photosynthesis equation again. We know it begins with carbon dioxide and water, uses light energy, and it changes those reactants into sugar and oxygen. Last time we looked at the light-dependent reactions, which split the water and gave us oxygen and some other high-energy molecules. So a review from last time, the light-dependent reactions take place here in the thylakoids. Light goes in and water goes in. Water gets split. Oxygen comes out, and we take the hydrogen ions and electrons from water to change ADP to ATP and NADP plus to NADPH. We also learned last time that these two molecules move over to the light-independent reactions, which are the topic of conversation today. During the light-independent reactions, carbon dioxide is going to go in and sugar is going to come out. And this is going to be powered not by light, but by these two high-energy molecules. During this process, something called carbon fixation occurs. So carbon goes from an inorganic source, carbon dioxide, and it gets incorporated into an organic molecule, which is glucose. The other thing you need to note is that the light-independent reactions are also called the Calvin-Benson cycle after the scientist who discovered them. They take place here in the chloroplast, and in particular, they take place in the stroma, which is that liquid that fills up the chloroplast. So that's where the light-independent reactions occur. The Calvin-Benson cycle occurs in steps, and the first step is carbon fixation. So carbon dioxide, which has one carbon, is going to diffuse into the chloroplast, into the stroma. And then it's going to actually combine with this molecule here, which is known as RUBP, which is ribulose biphosphate, but it's called RUBP for short. And it's going to combine with the help of an enzyme called Rubisco. So the carbon from the carbon dioxide is going to combine with RUBP. And together, they're going to form this molecule, which is a 6-carbon molecule that is super unstable. And this 6-carbon unstable molecule is going to break apart because it is unstable. And what is it going to break apart into? It's going to break apart into something known as phosphoglycerate right here, but it's called 3-PGA for short. So that 6-carbon molecule split into 3-PGA, and this is the part of the Calvin-Benson cycle known as carbon fixation because we took carbon dioxide and we changed it into this 3-PGA. The second step of the Calvin cycle is called reduction. So we're going to start with that PGA, that 3-PGA that we made in the previous step, and we are going to reduce it into something known as G3P for short, but the long version is phosphoglyceraldehyde, but G3P is much more useful and it's shorter. In your book, they abbreviate it as PGAL, but G3P, PGAL, it's the same thing. And what happens here is that each of these PGAs is going to get something from ATP and NADPH. So these came from the light-dependent reactions, and this is why they're so important because the ATP and the NADPH are going to help us go from PGA to G3P. How are they going to do that? The ATP is going to give this 3-PGA a phosphate, and the NADPH is going to give it some electrons and hydrogen to help reduce the PGA and form this G3P over here. So we're still working with a 3-carbon molecule. The next step of the Calvin-Benson cycle is known as carbohydrate formation. So most of these G3Ps, these 3-carbon molecules, aren't going to be used to make sugar at all. Instead, they're going to continue the cycle and they're going to help us make more of that RUBP, which is something that we needed in the beginning of the Calvin cycle. It's going to help regenerate that. But some of that G3P is actually going to be used to make glucose, which is a good thing because that's the point of this cycle. So what happens here is that 2 G3Ps are going to combine and they're going to help us form glucose. To actually get one molecule of glucose, we actually have to run this Calvin cycle 6 times. Okay? So let's say if we start with 6 carbon dioxides, we're going to form 12 G3Ps and we can make 2 glucose molecules, and the rest of those G3Ps, so the 10 extra ones, are going to help regenerate RUBP. So again, the part that we're talking about right now is carbohydrate formation. We take the G3Ps and we use them to form 6-carbon sugars called glucose. The last step is called regeneration. So we made some glucose and we're left with some G3P, and these G3Ps are going to be used to help us make more RUBP so that we can run the Calvin cycle again. And that RUBP can then go accept more CO2 and we can make more sugar. In this process, ATP is also required, which again came from those light-dependent reactions. So the last part is called regeneration, where the G3Ps are going to be used to make more of these RUBPs, and we can start the process over again. So here is another graphic showing you the same thing that I just showed you. It just runs in the opposite direction. We have your carbon dioxide going in. We formed some PGA, right? This was called carbon fixation. And then we made some PGAL. This part was called reduction. This part required ATP and NADPH from the light reactions. Here we made some carbohydrates in the form of glucose from the PGAL coming together. And then the rest of the PGAL went on to the regeneration phase, which required ATP again from the light-dependent reactions to regenerate that RUBP so that more glucose could be made and more cycles can be run through the Calvin-Benson cycle. Okay, stomata. So plants are covered with this waxy cuticle layer, and that is supposed to help the plant keep them from losing water because they really need that water for those light-dependent reactions, and they don't want it to evaporate. So they're covered with this wax. The only problem is that gases, oxygen and carbon dioxide, can't get through this waxy cuticle. So what the leaf has, if we zoom in really close to the leaf, are these openings called the stomata, which the leaf can pull closed or push open. And when it's open, then CO2 can come into the leaf and oxygen can come out. However, there's a problem because on hot, dry days, the plant is going to close its stomata so it doesn't lose water. However, the problem with that is that it can't get carbon dioxide then and it can't get rid of oxygen. So gas exchange comes to a halt. A consequence is that CO2 levels fall down inside the leaves and oxygen levels rise, which is not too good for photosynthesis. And what occurs in this situation is something known as photorespiration, which is really bad for a plant. And that is not regular cellular respiration. That is a wasteful pathway. And it occurs when that enzyme we learned about, Rubisco, instead of acting on carbon dioxide, it acts on oxygen and is super wasteful and not helpful to the plant. So there are different types of plants that have different ways of dealing with this situation. And the first type of plants include the C3 plants. And C3 plants include most plants, rice, wheat, soybeans, all trees. And these plants basically have no special adaptations to photorespiration. In hot weather, they close their stomata and they suffer. All that oxygen builds up in them, carbon dioxide goes down, and they go through photorespiration. And this is really bad for the plant. It results in lower crop yields, and it's just inefficient for the plant. It can grow less, and it will thrive less. So these are C3 plants. They don't do so well in this type of weather. The second type of plants are C4 plants, which are adapted to these extreme weather conditions, and they include bamboo, switchgrass, and corn. And they can minimize photorespiration because they fix carbon twice in two different cell types. So first, the cells will take in the carbon dioxide molecules, and they're going to fix them into these weird little 4-carbon compounds. And those 4-carbon compounds are going to get taken to a second type of cell where the Calvin cycle actually happens. And here, the 4-carbon compounds are going to break back down into CO2, and then carbon fixation is going to happen a second time, and we're going to run through the Calvin cycle here. The reason why this is good is because now when Rubisco is acting upon the molecules, there's a lot of carbon dioxide here and no oxygen because we broke up this process into two different steps. So C4 plants are adapted to this type of climate. The second type of plant that is adapted is the CAM pathway plants, and these include cacti and pineapples. And they minimize photorespiration by fixing carbon twice as well, except they do it at different times of day. So they carry out carbon fixation at night, and they fix carbon into 4-carbon acid, which is stored for later. Then during the day when it's really hot and the stomata are closed, they take that acid out of storage and they break it back into CO2, and that's when they run the Calvin cycle. So again, that Rubisco has a bunch of CO2 around it and it doesn't bind oxygen and it doesn't cause photorespiration. So these are how some plants can be adapted and use the Calvin cycle in different ways to minimize a harmful process known as photorespiration. All right, so as a treat, I'm going to show you a little video. And what you're going to see here, right here, this is a stomata inside of a plant cell. Look at what's happening to the stomata. You like my song choice? So this plant here is minimizing water loss, but also it is not having this gas exchange take place right now. Okay, so I hope that was helpful. I hope that was a useful introduction to the light independent reactions. Thank you so much for watching.